Overcoming MHC downregulation in cancer therapy by modifying mRNA vaccination. In the following, I would like to discuss one of the most important immune escape mechanisms of tumor cells, namely, the downregulation of MHC molecules, which reduces antigen presentation and immune recognition by T lymphocytes. My focus is not only on describing the problem but also on how mRNA vaccines can overcome this hurdle to ensure more effective tumor therapy. Causes of MHC-1 downregulation. The causes of MHC-1 deficiency in tumor cells include MHC mutations, such as those affecting beta-2 microglobulin, the loss of heterozygosity and HLA genes due to defective DNA repair mechanisms, or defects in antigen processing and presentation, for example, due to mutations in the transporter TAP1 and 2. Epigenetic silencing mechanisms such as histone deacetylation and DNA methylation may also contribute. Additionally, the tumor microenvironment is characterized by hypoxia and a high concentration of immunosuppressive cytokines such as TGF-beta and IL-10. As a result, gamma interferon production is low, which normally induces the expression of MHC-1. Consequences of MHC downregulation. Without the interaction between MHC and the T-cell receptor, immune recognition by CD8+ T cells is absent, preventing their activation. This also reduces the infiltration of other immune cells. Tumors deficient in MHC-1 have shown high therapy resistance to immune checkpoint inhibitors and mRNA vaccines. Alternative pathways for MHC-1 negative tumor cell recognition and destruction through NK cells. Natural killer recognize MHC-1 negative tumor cells through additional immune receptors. Self-recognition involves various interactions between NK cell receptors and ligands on tumor cells. When these ligands are defective, NK cells' cytotoxic activity is enhanced. Due to genomic instability and oxidative stress, tumor cells also express so-called stress ligands. NK cells recognize these via stress ligand receptors. Additionally, tumor cells express FAS and trail receptors, which are activated by ligands on NK cells, leading to caspase-mediated tumor apoptosis. Immune escape mechanisms against alternative pathways. Tumor cells can evade this self-recognition by shedding their stress ligands, expressing immunosuppressive cytokines, or expressing non-classical MHC molecules such as HLA-E or HLA-G, which inhibit NK cells. mRNA vaccine therapy and immune modulation in MHC-1 negative tumors. mRNA vaccines provide the opportunity to enhance immune responses in tumors with low MHC-1 expression by incorporating additional mRNA sequences. For example, introducing IL-2 mRNA can increase gamma interferon production over the course of therapy, thereby inducing MHC-1 expression. IL-2 also enhances the expression of stress ligand receptors in tumor cells, leading to increased activation of CD8+ T cells and NK cells. In several tumor cell lines, this approach has been shown to partially compensate for MHC-1 deficiencies. Further therapies to enhance gamma interferon production and MHC-1 expression. Gamma interferon concentration can also be increased by administering recombinant gamma interferon or by inducing its production with IL-12. IL-15 can enhance the expression of stress ligand receptors on NK cells. Additionally, immune checkpoint inhibitors targeting PD-1/PD-L1 and CTLA-4 can increase stress ligand receptor expression. Furthermore, DNA methyltransferase and histone deacetylase inhibitors can restore gamma interferon production and enhance NK cell activity. Radiation therapy can also increase MHC-1 production in tumor cells, as irradiated cell particles cause DNA strand breaks in neighboring cells, which in turn stimulate gamma interferon production.